Today's video is going to focus on the key concepts in cell biology of the cell theory, the functions of life, magnification, and the surface area to volume ratio. Beginning with the main principles of the cell theory, the first principle is all living things are composed of one or more cells. That is, cells are the building blocks of all organisms. The second principle is cells are the smallest unit of life. That is, cells are the most basic unit that are able to carry out all of the functions of life, which we will get to later in this video. The third principle is cells arise from pre-existing cells. That is, cells do not spontaneously generate. While modern cell theory also includes additional statements, concepts still hold true. Questioning cell theory by the use of atypical cells as examples. So most organisms conform to the cell theory. However, there are some organisms that do not conform to the cell theory, and we will explore those now. The first is the striated muscle cell. The striated muscle cell is because of muscle cells fusing together to form long fibers. Additionally, this fusion of muscle cells results in the cells having multiple nuclei. These two of the muscle cells fusing challenge the ideas that cells are small and only contain one nucleus. The second atypical cell is the giant algae known as the Acetabularia, and this atypical cell is a very large unicellular organism, as seen in the photo here, and it actually ranges in size from 0.5 centimeters to 10 centimeters in length. Additionally, it is a rather complex unicellular organism with many parts, namely the cap, the stalk, and the rhizoid. Together, these challenge the idea that cells must be small in size and simple in structure, as according to the cell theory. The final example of the atypical cells is the aseptate fungal hyphae. So fungi normally have a septa in their hyphae, as seen in these here. However, aseptate fungi do not have septum. As a result, they have a continuous cytoplasm and are therefore multi-nucleated cells. They are also not discrete cells as a result of this. This challenges the idea that cells only have one nucleus and the idea that living are composed of discrete cells. Now we will look at the functions of life. The functions of life are able to be carried out by a single living cell. This explanation of the function of life will be also illustrated in the following slide with two organisms be explored. So the first factor in life is metabolism. The metabolism is the web of all enzyme-catalyzed reactions in a cell or organism. Secondly, response. This is the reaction to a stimulus or change in the external environment. Next, homeostasis. This is the maintenance and regulation of internal cell conditions. Growth. This is the increase in size or shape that occurs over a period of time. Reproduction. The production of offspring to pass on genetic information to the next generation. This can either be sexual or asexual. Excretion, the removal of waste products from metabolic activity and other unimportant materials. Nutrition, this occurs by feeding either by synthesis of organic molecules, which is autotrophic, or the absorption of organic matter from the environment, which is heterotrophic. A mnemonic to remember this is Mr. H. Wren. As mentioned, the functions of life can be illustrated through the organisms of the Chlamydomonas, which is an autotroph which produces its own food, and a paramecium, which is a heterotroph which feeds on food found in its environment. Beginning with metabolism, both the paramecium and the Chlamydomonas metabolic reactions are catalyzed by enzymes and take place in their cytoplasm. In terms of growth, the paramecium consumes food, it enlarges, and once it reaches a certain size, it will divide into daughter cells. Whereas for the Chlamydomonas, The production of organic molecules during photosynthesis and absorption of minerals causes the organism to increase in size. Similarly, once it reaches a certain size, it will divide into two daughter cells. In terms of response, for the paramecium, the wave-like action of the beating cilia helped to propel the mecium in response to changes in the environment, which could be towards warmer water or away from cooler temperatures. For the Chlamydomonas, it senses light changes in its environment using its eye spot, and then uses its flagella to move towards a brighter region to increase the rate of photosynthesis. In terms of homeostasis for the paramecium and the Chlamydomonas, both maintain a constant internal environment by collecting water in their contractile vacuoles and expelling it through the plasma membrane. This process is known as osmoregulation and helps them to maintain their water balance. In terms of nutrition, the paramecium is a heterotroph. That is, it engulfs food in its vacuoles where digestion takes place. The soluble products are then absorbed into the cytoplasm of the cell, and it feeds on microorganisms such as bacteria, algae, and yeast. The Chlamydomonas is an autotroph. It uses its large chloroplast to carry out photosynthesis to produce its own food. In terms of reproduction, the paramecium can carry out both sexual and asexual reproduction, although asexual reproduction is more common and is known as binary fission. Similarly, the Chlamydomonas can carry out both sexual and asexual reproduction, and when it reaches a certain size, it reproduces either by binary fission or sexual reproduction. Finally, excretion in terms of the paramecium. The digested nutrients from the food vacuoles pass into the cytoplasm, causing the vacuole to shrink. When the vacuole is with its fully digested contents reaches the paramecium's anal pore, it ruptures, expelling its waste into the environment. For the Chlamydomonas, it uses the surface area of its plasma membrane to excrete its waste products. In terms of cell biology, magnification is another key concept. Some common units of magnification is 1,000 nanometers is equal to 1 micrometer. Micrometers is equal to 1 millimeter. Therefore, something that measures 50 millimeters long is 50,000 micrometers and 5 million nanometers. The formula for magnification is magnification equals the size of the drawing divided by its actual size. This can be illustrated through this diagram, which is I am, where I is the image/drawing size, A is the actual size of the image, and M is the magnification. Now, the image versus the actual size of the image. The actual size of the image is normally what the scale will say on it, whereas the image or drawing size is what would be measured using a ruler. Another point is that when calculating magnification, all units must be the same. That is, if the actual size of the image is in micrometers and it is measured in millimeters, these must be converted to the same measurement. As an example of magnification, for instance, say a bacterial cell is viewed under a microscope. Its actual size is 4 micrometers. It would look something like this, and we would measure that, and we would measure it to be 50 millimeters. Therefore, since the magnification equals the size of drawing over the actual size, I'm converting the 50 millimeters into micrometers, which is 50,000 micrometers divided by 4 micrometers, gives a magnification of 12,500 times. Finally, the final core concept in this video is the idea of the surface area to volume ratio. So the reason cells cannot grow indefinitely lies in its surface area to volume ratio. This is because volume increases at a much faster rate than surface area. As a cell grows, its volume increases by a power of 3, whereas its surface area increases by a power of 2. Therefore, as a cell grows, the surface area to volume ratio is decreased. To survive, cells must import molecules and expel waste through their plasma membrane. If the cell's surface area is too small compared to its volume, insufficient molecules can get in and insufficient waste can get out. By cells dividing, they restore a high surface area to volume ratio, in turn allowing for optimal movement of materials in and out of the cell. So thank you for watching this video. I hope you found it useful.